After studying this module, you shall be able to know about the protein purification, learn the background factors in protein purification and extraction. Introduction. To achieve purification, the protein has to be released from its biological matrix and removed selectively from other proteins by an appropriate fractionation procedure. The purification strategy adopted in this fractionation process is dependent upon the factors quality, quantity and cost. Protein differ in their sensitivity to denaturation during the extraction and purification processes. In particular, they differ in their sensitivity to elevated temperatures, to the presence of detergents and heavy metals. These differences are commonly exploited in the purification strategy for a particular protein, and techniques based upon them commonly feature in the early stages of the purification process. The solubility of native protein is influenced by pH, by the addition of salts such as ammonium sulphate, and by the presence of organic solvents such as acetone and butanol at low temperatures. All the proteins are susceptible to proteolysis by the action of endopeptidases and exopeptidases, which are present in all cells, especially in lysosomes, but the magnitude of this susceptibility varies from one protein to another. While denatured proteins are all equally vulnerable to proteolysis, native proteins are not. Precautions therefore have to be incorporated into the purification strategy to minimize proteolytic degradation. Protein extraction. The cellular location of the protein to be purified determines the procedures that can be used to obtain an extract containing the protein in soluble form. Usually, mutually the extraction and purification procedures should be carried out in a cold condition, frequently at 0 to 4 degrees, excluding certain proteins. An ice box or alternative cooling arrangement is generally required for sample refrigeration. The abstraction process differs as per the category of sample and physicochemical functions of the protein. The initial step is to rupture cells or tissues. This process is applied to obtain more integral protein. Generally, the animal cells and muscles are soft and easy to disruption, so procedures can be applied. For bacteria, fungi and maximum plant cells that has rigid cell covering, further active processes are needed. Extraction buffer or solvent is also significant. Buffer with appropriate concentration, ionic strength and pH is applied for withdrawal of water soluble protein. In certain cases, insignificant detergent or other suitable detaching material is added into the extraction buffer. Protease inhibitors are generally essential to avoid obliteration of the extracted proteins by proteases, from the fact that the protein extract acquired from subcellular constituent has impurities not as much as that acquired from entire cell and will be at ease to be decontaminated. So, detachment of subcellular constituents may be done before the protein extraction. Rupturing of the cells must be accustomed to get integral subcellular components. The needed cell element is acquired by centrifugation in presence of suitable circumstance. Extraction of water soluble protein from animal cells and tissues. Components of fragile unicellular tissues, for instance animal blood cells, which comprises of proteins, can be easily removed by the application of hypotonic buffer solution. If the sample comprises various forms of cells, separation of cell types prior to the extraction will make the purification easier. This osmosis based technique is also applied for animal cells developed in culture media. In certain cases, a minor surfactant is comprised in the extraction buffer. Application of thaw freeze sequence or minor mechanical agitation or sonication may be done to help disrupt the cells and also in dissociation of cellular components. For multicellular soft tissue, a hand homogenizer or an electrical one with optimum dimension is a noble choice. The small crusher with a microcentrifuge tube is also commercially accessible for the sample with a small volume. Equipments with stronger breaking power like a blade mixer are required for withdrawal of proteins from rougher tissue like animal muscle. Cell rupture by nitrogen decompression is alternative process for animal cells and some plant cells. In this technique, a great amount of nitrogen are mixed in the cells in higher pressure in a vessel. Although the pressure is sharply unconfined, dissolved nitrogen gets converted into bubbles. Cells get ruptured up due to expansion of bubbles. By this technique, intact organelles can also be obtained. Protein purification. To refine any protein, numerous separating procedures are applied depending on physical and chemical properties of the protein. The purification procedure can be explained as given. Step 1, crude extract of protein. Step 2, separation into fractions. Step 3, detection of the desired protein, recognition by size, activity or property. Step 4, separation of the purified protein. Step 5, detection of the desired protein, detection by size, activity or property. Step 6, determination of the purity, electrophoresis. It is not necessary to follow all steps or all techniques for every protein. For example, myoglobin is easily detected by its red color or identified by applying UV visible spectrophotometric technique. So, no requirement to use any sophisticated procedure. Principles of separation techniques are as follows. Separation based on solubility. The hydrogen bonding with water molecule and charge present on hydrophilic protein determines the solubility of those proteins. At the isoelectric pH, total charge on a protein is 0, and that is why the molecules of protein easily get combined and then precipitate out. The technique is known as isoelectric precipitation. In case the pI is unknown, proteins may get precipitated by high concentrations of some cells, thereby resulting in the destruction of hydrogen bond between water molecule and protein. The technique is well known as salting out or salt precipitation. Ammonium sulfate is generally applied as it has great ionic strength. By the gradual addition of salt, proteins present in crude extract can be separated into a number of fractions. The precipitated proteins are redissolved and applied for advanced stages. Arrangement of the two procedures is applied for precipitation of certain protein. Separation based on size and density. The method comprise centrifugation, dialysis and molecular filtration. A high speed centrifugation about 10,000 to 20,000 G is usually applied to eliminate cell remains and large organelles out of raw protein extract if the preferred protein is a water soluble molecule. The supernatant is then applied for advanced refinement steps. To know the location of protein in cell, the cell rupturing should be an easy going technique to acquire integral organelles and subcellular elements. The subcellular element comprising organelles are then fractioned to applying various centrifugation. The subcellular element comprising organelles are then fractionated by applying variance centrifugation. In differential centrifugation, the subcellular element are alienated as per their sizes into fractions by the stepwise increase of the centrifugal force. Density gradient centrifugation is a technique to isolate particles containing protein masses by their concentrations. Liquid density gradient may be preformed prior to their application for the isolation. Several resources can be applied to create the gradients like a sucrose ficol. In addition, the liquid density gradient can be made in the course of isolation process. By this approach, the sample is coated on topmost of an appropriate concentration of cesium chloride. The strong centrifugation force of ultracentrifuge causes migration of cesium chloride into the bottom of the tube. The liquid density gradient is then created by the gradient concentration of cesium chloride. The density gradient is also spontaneously generated during centrifugation by using percol, that is polymer coated silica particle. Dialysis is a widely used method. The method is simple but time consuming because the separation depends on diffusion. The sample is kept in the dialysis carrier made by a tube formed from semi-permeable membrane. Depending on a commercially accessible dialysis tube, only smaller molecules which have masses less than 10 kilodalton is detached out of the sample to the surrounding medium. So, this procedure is generally applied to eliminate salts from the solution of protein. Dialysis is also applied to quantify protein solution. Water molecules are eliminated from the inside of dialysis container by applying a hydrophilic polymer such as polyethylene glycol. Chromatographic technique. Chromatography is the powerful method for detection and purification of biological substances. The principle of chromatographic separation is distribution or partition of separated molecules between two immiscible phases called mobile phase and stationary phase. For example, paper chromatography is called according to the material used as the stationary phase. Thin layer chromatography or TLC and column chromatography are entitled by the physical forms of the stationary phases. Gas liquid chromatography has gas mobile phase and liquid stationary phase. By using mechanisms of separation, chromatography is classified as adsorption chromatography, partition chromatography, size exclusion chromatography, ion exchange chromatography and affinity chromatography. Column chromatography is the most popular method for purification of proteins. The conventional column chromatography is performed under low pressure. The mobile phase runs by the stationary phase in the column by the gravity or by low pressure pumps. Thus, it can be entitled low pressure liquid chromatography or LPLC. High performance liquid chromatography HPLC is a conventional form of column chromatography. High pressure pumps, good quality constituents applied as stationary phase and sensors with great sensitivity are applied to modify the isolation and to diminish the operating time. HPLC is currently very prevalent for recognition and purification of biological molecules. The other chromatographic methods are fast protein liquid chromatography or FPLC, capillary chromatography, reversed phase liquid chromatography, etc. FPLC is similar to HPLC, but its working pressure is lesser than that of HPLC. LPLC, HPLC and FPLC are extensively applied for decontamination of proteins. Other separation techniques. There are some other minor methods used for purification of protein such as electroelution of the protein separated by STS page. The discolored protein band is taken out of the gel. The gel slices with an appropriate buffer are packed in a dialysis container. The bag is placed in the buffer between two electrodes. The protein is then electrically eluted from the gel slices. One can make a simple electroelution cell or can take any commercially available apparatus. STS is removed from the protein by dialysis in the attendance of a non-ionic detergent. Only a small amount of protein is obtained by electroelution and it is possibly denatured. Most proteins have been normally purified by the above techniques. To acquire integral hydrophilic protein, the decontamination procedure chiefly includes salting out and chromatographic techniques, though certain proteins can be decontaminated by simply specific procedures. For example, molecular filtration in combination with sodium chloride solution was successfully used for purification of glutamine synthetase. So, the simple specific procedures may be made for purification of some protein if the protein has the specific property. Decontamination of glutamine synthetase out of chick retina was done by applying molecular filtration in the presence of 0.7 molar sodium chloride. The crude extract and the purified protein were separated on STS page and then dye stained. Lane M molecular mass marker, lane 1 for crude protein extract, lane 2 for purified enzyme. Detection of protein. Purification procedure comprises numerous steps and is laborious. Therefore, it is necessary to be certain that the desired protein is present in the crude protein extract in all purification steps. Used for the protein with identified molecular mass or size, the most adaptable technique is gel electrophoresis, particularly STS polyacrylamide gel electrophoresis. Determination of enzymatic activities or other properties such as absorption spectra are also widely used. In certain examples, the refined technique LC-MS is implemented for identification of the protein after separation by electrophoresis. Electrophoresis, it is the most widely used technique for separation, detection and determination of the purity of protein. It is sometimes used for protein purification. The technique is based on migration of charged proteins in an electric field. Electrophoresis of proteins is generally conducted in a gel medium, mostly polyacrylamide. The widely used polyacrylamide gel electrophoresis are sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. STS page is usually applied for determination of clarity and molecular mass of protein. This versatile method is reproducible and cheap. For the principal, sodium dodecyl sulfate in the system binds to most proteins in amount approximately proportional to the molecular mass of the protein. Therefore, every protein has a like charge to mass ratio and migrates proportional to its molecular mass. The extensively applied STS page is the lamellae procedure as well as its improved procedures that involve of the uneven buffer system. In this buffer system, there are two parts of polyacrylamide gel, stacking gel and separating gel. There are variances in arrangement, concentration and pH amongst electrode buffer, assembling buffer and splitting buffer. The usefulness of the buffer stem is stacking effect which occurs in the stacking gel. The electrophoresis set consists of the upper and lower chambers which are filled with an electrode buffer. The gel is polymerized in the space between two glass plates and then connected between the two chambers. Protein samples are loaded in wells at the top of the gel. Protein molecules will migrate into the gel under the electric field. For STS page, the proteins migrate from cathode to anode. Separation of the proteins occurs in separating gel. STS page can be applied to determine molecular mass of the protein by using a calibration curve between relatively mobility and long molecular mass of standard protein. There are the various procedures of STS page including urea SDS page, trypsin SDS page, etc. The other type of polyacrylamide gel electrophoresis are also available, such as acid urea page, native page. Although SDS page is commonly used, some proteins may need specific page for the detection. The proteins separated on the polyacrylamide gel can be visualized by many types of staining. The discoloration with dye, particularly Kumasi Brilliant Blue R250, is extensively applied as it is cheap and efficient. Staining with some other dye and silver staining are also available for specific purposes. Now, let us summarize what we have learnt in this module. Proteins differ in their sensitivity to denaturation during the extraction and purification processes. In particular, they differ in their sensitivity to elevated temperatures, to the presence of detergents and heavy metals, and to extreme of pH. Usually, mutually, the extraction and purification procedures should be carried out in a cold condition, frequently at 0 to 4 degrees, excluding certain proteins. An ice box or alternative cooling arrangement is generally required for sample refrigerating. To acquire integral hydrophilic protein, the decontamination procedure chiefly includes salting out and chromatographic techniques, though certain proteins can be decontaminated by simply specific procedures. Determination of enzymatic activities or other properties such as absorption spectra are also widely used. In certain cases, the refined technique LC-MS is implemented for identification of the protein after separation by electrophoresis. SDS page can be applied to determine molecular mass of the protein by using a calibration curve between relative mobility and log molecular mass of standard protein.